Hi YouTube, my name is Michael Berenger. I'm a medical doctor and a full professor of sports medicine and exercise physiology at Goethe University, Frankfurt, Germany. Welcome back to our muscle physiology series. In the last episode, we explored the three types of muscle tissue and what makes skeletal muscle unique. Today, we're going to zoom in, literally. We'll break down how skeletal muscle is organized from the whole muscle you train down to the microscopic sarcomeres where movement truly begins. Along the way, we'll also take a closer look at the connective tissue that not only supports muscle structure but plays a vital role in force transmission. So let's do this. One of the most fascinating features of skeletal muscle is how it's built. Skeletal muscle has a beautiful layered structure. At the top is the whole muscle, the macroscopic structure you can actually see, touch and of course train. But that muscle is made up of bundles called fascicles, and each fascicle contains many long cylindrical muscle fibers, also known as muscle cells. This is often misunderstood. Many people think muscle fibers and muscle cells are something different, but they are actually the same thing. Inside each fiber, there are myofibrils, intracellular structures responsible for contraction, and inside those are the sarcomeres, the contractile engines. And each layer is surrounded by connective tissue that maintains alignment and provides structural integrity. So let's have a closer look at the connective tissue layers. We distinguish three distinct layers. First of all, there is the epimysium, and this surrounds the entire muscle and anchors it to the surrounding tissue and tendons. It also connects to the deep fascia, forming an interface between muscle and surrounding compartments. Then we have the perimysium that wraps around each fascicle and serves as a pathway for blood vessels and nerves. And finally, there is the endomysium. This surrounds each muscle fiber and helps to distribute capillaries and transmit force laterally. So these aren't just passive wrappings. Together they form a continuous three-dimensional collagen network called the intramuscular connective tissue, or IMCT. This network integrates with tendons and even deep fascia to distribute force throughout the muscle. And mechanically, these layers are involved not just in tension, but also in shear force transmission, which helps synchronize and distribute force across neighboring fibers, providing mechanical stability and protecting against overload. Different layers contain different types of collagen. Type 3 in the endomysium for fine flexibility, and type 1 in the perimysium and epimysium for tensile strength. Let's talk about fascicles and muscle fibers. A fascicle is basically a bundle of muscle fibers. Imagine a pack of spaghetti wrapped in plastic. Each fiber is a single muscle cell, and here is the amazing part. They can be up to 15 to 30 centimeters long in humans, so they are some of the longest cells in our body. Since each nucleus governs a certain volume of sarcoplasm, known as the myonuclear domain hypothesis, each fibers contain hundreds of nuclei per millimeter, distributed along its length, and they are located at the side of the fibers. Within each fiber, you'll find a dense, repeating internal architecture. First, there are the myofibrils, long rod-like structures arranged in parallel. These are the actual contractile elements of the cell, and each myofibril can be divided into repeating units called sarcomeres, which give muscle its striated appearance and generate force. Surrounding the myofibrils is the sarcoplasmic reticulum, or abbreviated as SR, a network of specialized membranes that form terminal cisternae and tubules designed to release calcium to initiate the contraction. Interwoven with the SR, there are the transverse tubules, or T-tubules, which are invaginations of the sarcolemma. These structures conduct an action potential from the muscle surface deep into the fiber, ensuring that the contraction signal reaches all myofibrils almost simultaneously. Together, the triad of T-tubules and SR forms a critical unit for excitation, contraction, coupling, the process that translates an electrical signal into mechanical movement. So let's talk about the sarcomere structure. Each sarcomere is bordered by Z discs, which anchor the actin, the thin actin filaments. You can think of the actin network as a kind of a passive scaffold. Between those thin actin filaments lies the thick myosin filament, the actual motor of contraction. During contraction, it essentially pulls itself along the passive actin scaffold using myosin heads that attach, pivot, and release in a ratcheting motion, so to say. Since the myosin filaments are anchored in the center and interact with actin filaments on both sides, they pull the thin filaments towards the middle from opposite direction. As a result, the Z discs at the ends of the sarcomere are drawn closer together and the sarcomere shortens. This is the fundamental mechanism of muscle contraction. Exactly how the myosin heads produce this movement, how they attach, pivot, and release is something we're going to explore in detail in the next video. Other proteins like titin and nebulin maintain the structural integrity and elasticity of sarcomeres. Titin spans from the Z discs to the M line and acts like a molecular spring, contributing to a passive stiffness and alignment. Nebulin, on the other hand, runs along the length of the thin actin filaments and acts like a molecular ruler. It helps define filament length and stabilizes the actin structure by binding F-actin, the filamentous form of actin polymerized form of individual G-actin monomers. Tropomyosin is another key protein associated with the actin filaments. It winds around the F-actin helix and, together with the troponin complex, regulates access to the myosin binding sites. In a resting muscle, tropomyosin blocks these binding sites. So to allow myosin to interact with actin, tropomyosin needs to be moved out of the way. We'll explore the exact molecular choreography behind this interesting mechanism in the next video. Let's briefly recap. Skeletal muscle is built in layers: whole muscle, fascicles, fibers, myofibrils, and finally sarcomeres. Each level is supported by connective tissue that helps align structures and transmit force. The sarcomere is the core contractile unit, and in the next video we'll take a closer look at how proteins like actin, myosin, and titin interact to generate movement. If you're enjoying this series, don't forget to like, subscribe, and leave a comment. Thanks for watching. See you soon. Bye-bye.